Hello DYBS students, uh, welcome to virtual classroom. I am Dr. Anirudh Bhagat. Today we are having one more lecture on parasitology, which will include uh, the parasite adaptation again, or their specificity, and basically the structural specificity. Now look at here the first slide. This is about the previous lecture. In that lecture we have already gone through the host specificity of parasites, which is divided into three parts, which is physiological specificity, structural and ecological specificity. We have already seen in previous lecture about the physiological specificity. So in that we have seen what is the physiological specificity, how it is compatible with the which is not compatible with the foreign host but compatible with the normal host, and what are the examples. So gut tapeworm examples we have seen. Why we have seen we have taken this example. Other than that, we have seen the Taenia solium example, why it cannot survive in dogs, why humans are only required. Other than that, we have discussed one more example related with the experimental setup, in which we have talked about the biting lice of the poultry and why it cannot survive on green heron. Okay, so and along with this we have already gone through one more part, that is called as ethological behavior, or sometimes it is called as ethological specificity. Okay, in which we have taken example of pork tapeworm and Taenia saginata and Taenia solium. So pork tapeworm and beef tapeworm. Okay, and based on that we have seen what are the physiological adaptations that has actually occurred within the parasite. It was related with the intercellular, then osmoregulation, anaerobic respiration. Today we will start with the another part, which is the structural specificity. Actually the sequence is a structural specificity, then physiological specificity. Okay, but I have taken physiological first and then structural. Today I am taking it. So what is this structural specificity? So this is more related with the what are the external adaptations that has occurred within the parasite. So if you look at here, the structural or morphological adaptation of parasite to its host is called as structural host specificity. Kind of adaptation of parasite towards its host. So whatever host it has, the parasite is adapted according to the host. Also, host specificity is a result of extensive specialization of parasite to one host, and adaptability for the other host is lost. As we have discussed earlier, that these parasites, they have evolved along with the host. So naturally they come maximum in contact with its definitive host, and if they are coming maximum times in contact, so naturally it will try to get adapted. So throughout the course of evolution, they have adapted themselves toward the host environment, because host is its natural environment. So remember, these parasites which are present in host in adult form, this adult form cannot survive outside the host, remember. So in their life cycle, some other form, for example larval form, can survive within outside the host, but once it is in definitive host, it gets converted into adult host, and once it is adult cannot come out of the host, and if it does, it will die, because it is not adapted for outer environment. Even it will not be adapted for any other animal also. Why it is so? Okay, so some examples. The first one is about the liver fluke, okay, which have to pass through stomach initially, okay, in order to reach the bile passage for further development. So how it enters into the human body, it in the form of cyst capsule. Okay, So first step, when it enters into the body of human, at that time it will enter into the stomach, and within the stomach there are gastric juices and its pH is 1 to 2. So the cyst capsule, it's to be protected against all these environment. So it is provided, the cyst capsule is provided as a protection against the digestive juice. Okay, so the liver fluke, the cyst capsule outer covering is so hard, or it is protective against all kind of juices. Another adaptation, if you look at modification, liver fluke body is leaf shaped. Okay, dorso-ventral pattern, and entire integument become thin. So it is very thin. It has to be present. This is called as liver fluke. It has to be present on liver. Now, other than liver, along with the liver, there are so many other things also present within the body. If it will be bulky, it will not fit in that area. So to slip through, or it should be present on the liver, it is necessary that its size should be dorso-ventrally flattened. Okay, and because of this, it remain within the liver. Okay, then you have another example, anisakid nematodes. Okay, it's a parasitic disease, okay, caused by anisakid nematodes. So it can invade the stomach wall or intestine of humans, right? And the transmission of this disease occurs when the infective larvae are ingested from fish or squid that human eat as a raw. There are many countries where people eat raw meat most of the time, especially the seafood. Okay, they will have a piece of fish directly, they may eat it. Squids, or you have heard about octopus, people directly eat the octopus, small octopus. Generally this is not found in octopus, but yes, raw meat, uncooked meat. Okay, so it will enter inside, and they are provided with a thick resistant integument, chitin like, which is first of all protects them against the gastric juices, and it is impermeable. Okay, so they may pass through to the intestine or somewhere and enters into the body. So this is one example. Another example is of Ascaris. Okay, Ascaris is provided with the anti-enzyme to resist the action of digestive enzymes which are present within the body. So for initial entry, at that time it will be digested by our stomach. So this should not happen. That's why they have some enzymes which is to be resisted, which resist the digestion within the stomach. Other than that, the structure of many tapeworms has been closely correlated with the villus structure of host. Villus means within the intestine. Okay, the villi that you say. So their structure is somewhat adjustable to that villi which is present within the stomach. So these are some of the structural specificity. Also there are some more examples, or you can say the adaptations. So this initial was related with the protection against the juices, so that kind of specificity they have. Then many nematodes living in the intestinal tract provided with the spinous integument to guard against the abrasive action of food and roughage passage in the gut. There will be lot of food, so they should not get, as you know, when food enters into the stomach, there will be continuous movement of the food. Because of the movement, there is a possibility that the food, solid food, it may damage the parasite. This doesn't happen. Okay, now remember why all these things are, why it comes into the structural specificity, because these things makes them stable within their environment. Okay, so spines are there. Same liver flukes have many spines and sensory papillae initially. Apical membrane will thrown into ridges and pits and the microvilli. Okay, the outer part of the integument contains secretory bodies which continuously released at the surface to renew the apical membrane. Okay, there is one, mainly the example is of the liver fluid C. sinensis, which was probably then intestinal parasite before it become a bile duct inhibitant. Okay, so they also have the spinous integument type part, and these are all the protective devices against the also these are all the protective devices against the human immune system. Okay, so they have to protect themselves against the human immune system also, other than the digestive juices or abrasions. Then there are some more structural specificity. For example, the best example, there are some parasites, ciliates, which are present in cecum of the horses, and there are from some flagellate in the hindgut of termites. I have already talked about this. The termites have, those are not parasite actually, some of them are parasite which will be present in the termites. Okay, so they are adapted in the cecum area itself, not in other part. They will present there for longer duration. There is one example of many times what happens, a single morphological character of parasite become limiting factor to that particular parasite. So we have seen parasites which will have only one definitive host, or they may have a primary and secondary host. Generally the second host is considered to be the intermediate host or a vector. Okay, but these parasites are adapted to the intermediate host at the different life cycle stage, and the adult form is adapted for a different life cycle stage with some another host. I have already talked about this, that liver fluid when initially enters, it is in the cyst capsule. Okay, so that life cycle stage is a different one, and when it become adult, and the adult one doesn't enter in somewhere else. So this is very important, but sometimes it become limiting factor. For example, the posterior sucker of leech, this is one example, Halobdella. Okay, it is adapted in shape and size to the ctenoid scale of flatfish solia. So ctenoids, we have seen different types of scales. We people have studied this different type of scales. Even practicals were also there in the I think in SY you have that practical different type of scales. Okay, so in that ctenoid scales, now the posterior sucker of this particular leech is somewhat you can say lock and key type, and it attaches itself to the ctenoid scale, but of that particular fish. But if that scale is not present, then it will not be able to attach. Okay, so this shows that because of this, it is specific towards the fish solia, but it cannot go to other host, even though it feeds on the fish itself, but it only goes towards the solia, or some of the fishes may which have ctenoid scales. Okay, because of its mouth part adaptation. So leech is unable to attach to other fish species. Okay, so they have a strict host specificity. Okay, this you should know, where strict host specificity. Now other than this this structural specificity, there are more modifications that also leads to the structural specificity. Now these modifications for attachment is related with for example here sucking organs. Okay, again example of liver fluke is there, which is two suckers present on ventral side of the body, anterior and posterior. In case of human tapeworm, it consists of either sucking tongue or groove are there. Okay, so for example, the tapeworm, the scolex, four large suckers are there. Outgrowth of this scolex called as bothridium. Okay, and each bothridium with also have the suckers. So there are some changes on that. Other than this, as you can see, there are some more adaptations. It is related with the hooks. Now these all adaptations are related with the intestine, and remember I have already told you that intestine if there is a difference in the intestine, parasite cannot survive. Okay, so these parasites, they cannot survive in carnivorous animals. Carnivorous animals do have parasites. It is not like that only herbivorous animals have a parasite. Their parasites are different and they are adapted to the carnivorous animals digestive system only. They cannot go towards the herbivorous animal digestive system. So this is very important. So if you look at the some of the example of again the structural specificity, presence of hooks, okay, which is around the anterior end. What these hooks do? They provide the attachment to the intestine. Okay, so intestine, which is not a smooth area, as just now I have told you, the villi are present. So to those villi, they should be able to attach themselves. Okay, so this hook provide with a series of teeth and are placed in the buccal capsule. So this is one part. Another thing, glands. I have already told you that some of the glands which produces anti-enzymes, but when it gets entered into the intestine, there are some other glands. So it secretes a liquid from unicellular gland to dissolve host tissue, thus making microscopic passage for the parasite. For example, Merasidium larvae of Fasciola hepatica. So this Merasidium larvae, when enters into the pila skin, so they have the glands which produces the enzymes which which allow the entry of this particular larvae into the fasciola, in the pila, and later from pila they may enter into the human body. That is a different thing. In hookworms, okay, there are glands in the buccal region which are supposed to have anticoagulative and histolytic properties. So anticoagulative property, they require blood, okay, and if continuous blood flow will be there, what will happen? There is a possibility clotting formation. So anticoagulative means it will not allow the blood to clot, okay, and histolytic properties. So this is a kind of adaptation again, because they want to survive there, they want to feed, and during the feeding itself this should not happen. I have given you an example, ectoparasite, that is of a vampire bat. That vampire bat saliva also contains some anticoagulative enzymes which doesn't allow the clotting of blood, because once you prick, it will have very sharp teeth, and when it start pricking the skin, blood will come out, it will start eating the drinking the blood, and there is a possibility that because of the clotting factors blood will clot. This should not happen, that's why it uses the saliva. Similar way here also, hookworms also have a similar kind of part. So these are some of the specificity which makes that particular parasite limited to specific host. Because if we take the hookworm and keep them somewhere else where they will not be able to do this, or not be able to penetrate the skin, then they will not survive. Similar with the Merasidium larvae, if that host is not present, or it will not be able to penetrate the host, it will not survive. So generally Merasidium larvae doesn't penetrate the direct skin of humans, they have to go through the pila, and that is their host specificity. Merasidium is not meant for humans. Merasidium larvae is meant for only, that stage is meant for only the pila. Okay, so this is all about the structural specificity. In next lecture we will discuss about the ecological specificity, and other than ecological specificities there are more specificities that we will cover in next lecture. Thank you.